Okay, so let's talk about the second part of this lecture, that is the pairwise sequence comparison. So we all know the proteins are evolutionarily related. Typically, an ancestor protein will have a so-called orthologs and paralogs. Ortholog basically means by this inheritance. That means that you have one gene, and in evolution, another species will also contain the same gene, but with this with some variations. And then you also see this so-called gene duplication. This happens very often in plants. That is either the genome or that gene is duplicated. So previously you have one gene, now you have 2 copies or more copies of these genes. So in all these cases, their sequence will be similar. And then there are also cases called fusion. That means that you have let's say, a new gene, but this new gene, part of it came from this gene, part of it came from another gene. It forms a mixed homology. And in all these cases, you actually can find such relationships by sequence comparison. So for protein study, the sequence comparison is the most important and the most fundamental operation, because it's the key to understand the evolution of the organism and also evolution of that particular gene. And then for sequence search, of course, you really need to use this sequence comparison. And then once you find a similar protein, if that protein has some known information, then you can transfer the information to acquired protein, either in terms of structure, function, evolution, or maybe sometimes we do it for design purposes. And also sequence comparison is the basis for other prediction methods. For example, protein localization prediction. We rely on sequence comparison. So here, I'm not going to talk about this algorithm behind it, but just talk about the basic idea of sequence comparison. So this so-called alignment approach. That is, if I have 2 sequences, you align them. And then to make sure that the same letter or similar letter, they will be aligned together. This is done through some scoring function. So the same letter we know, for example, F, they are phenylalanine. So they are aligned together. But D and E, they're not same amino acids. But both of them have negative charges. So we think they are similar amino acids. That means that when we align them, we give some reward. In other cases, we don't give any award or even give some penalty. And then we need an algorithm to do it. Of course, this is a dynamic programming or heuristic search. If you just use it as a biologist, you don't have to understand how this algorithm works. But the basic idea, you just to optimize this score. And then another important thing that so-called confidence assessment. That is given this alignment with a match, mismatch, substitution, gaps, etc. How good the alignment is. So if you just see the score, it's not that helpful because it doesn't provide you so-called confidence level, how trustworthy or how useful this alignment is to study biology. So before confidence assessment was developed, the alignment methods actually, there were many, but not widely used in biology. But after confidence assessment, it actually proposed for sequence alignment. It's actually by this giant in physical methods, Sam Carlin. It really laid the foundation for biological applications Because once you have the confidence assessment, you know how useful this alignment is, how much trust you can place on this alignment. So this is actually very critical. And then the alignment that you can have this so-called global alignment or this local alignment. Yeah, the global alignment basically means that you align both sequences end to end. You consider every portion of it. And then local alignment basically means that you only need to align part of it. So let's say this part alignment is very good, and then other parts are not very good. Then you can ignore them. You do not penalize the alignment by those regions. You only calculate score for this part. So this is a local alignment. And then in both cases, you can get a confidence. Of course, this confidence is more meaningful for local alignment. Here, typically, people use this so-called expectation value or E value for measuring the confidence. And then local alignment is more widely used to study biology. Global alignment is also used sometimes. For example, when you build a 3D structure model, people tend to use global alignment more than local alignment because you want to match the entire structure instead of just, let's say, one alpha helix of that structure. So for alignment, there are just tons of methods. But typically in any biomedical field, initially you got a lot of methods, but later you'll be settled to a handful of tools. And typically one tool is dominant. In sequence comparison, the dominant tool is BLAST. So the dominance is really amazing. So this tool was published in 1990. And this paper was cited tens of thousands times. Yeah, some people say this is one of the most cited paper in scientific history. So that really demonstrates the impact of this tool. So here is the link, which I'm going to do the demo. And then it just perform the local alignment to find identical or similar sequence in a short segment. But this alignment can be done iteratively through the so-called SciBLAST. SciBLAST basically means a position-specific iterated blast. So you run BLAST multiple times. Each time you construct position-specific score matrix to replace the query sequence for the next round of search. The idea that you build this so-called profile, that is, you find in this alignment which position are conserved. And then more conserved residues, you give a higher weight when you calculate score. Less conserved, you give less, or if you violate a conserved pattern, you give a very negative penalty. So that's kind of the idea behind the SciBLAST. And then you run this several times. Sometimes you can just do, let's say, once. Usually you do 3 to 5 times to get significant hits. If you run more, it may not help. Because this tool is very sensitive. You can get more hits than BLAST, but it can actually deviate from the biological patterns. It gives you some sequences without any biological relationships. This is happened because of this random fluctuation. So let's say if a fluctuate to a certain pattern, when that pattern actually noise, it's not the real biological information. And then you amplify that noise, then you tend to branch out to the evolutionary path. It goes to another path, and then you find the proteins not directly related to your query. So that's why you don't want to search many times. And also you take a grain of salt The SciBlast gives you more hits, but those hits you need to manually evaluate. And also, in this case, the e-value can provide a good guidance, but not a highly reliable guidance, because sometimes it could be misleading. So more manual curation would be needed for SciBlast results.